हमने ना हमने पूरा भेज दिया ना सर, उसी में से कॉपी करके लिख
सही ना, भेज दो आगे।
हाँ
पर ये सही चल रहा है ना, देखिए सही है।
हाँ दोनों ही सही है सर।
ये थोड़ा सा
सर ने बस वहाँ से यहाँ तक शेयर कर दिया, इसमें से कॉपी करके।
अच्छा ये ऐसे तक है,
हाँ
ठीक है, उसी को कर देते ना। अभी ये किसने किया है?
कोडिंग करने के लिए।
ये किसने भेज दिया?
ये तो हमने भेजा ना।
SOP पे किसने भेजा? पोस्ट ठीक है, ठीक, हाँ कर दिया कर दिया, थैंक यू। शारदा वाली पे और गूगल मीट भी है सर? इधर ये दिया सर Hello, ma'am.
Good morning, sir. Professor Siddiqui, Dr. Ankita here.
Hello, yes, good morning, Ankita. How are you?
Sir, I'm fine. Hope you are doing well.
Yeah, Alhamdulillah, it's good.
Pleasure to
Thanks
see you
for,
here.
yeah, nice to see you all again. And I'm really congratulating all of you for organizing this 5 day workshop regarding FDP. And I hope the last 2 days, what you have done is fruitful and useful for the community. So, do you have my this presentation, or I should start from my end?
Sir, I do have it, as you say.
Okay, I'm trying first, then let you know if it is not working. Sometimes.
Okay, sir.
Good morning, Dr. Nasir.
Yeah, yeah. Good morning, Dr. Nair. How are you?
Bas fine. Really good to see
Yeah,
you after a long time.
yeah, yeah. Thanks to you too for remembering me after a long time.
These are the times we will remember our close ones.
Yeah, yeah, yeah.
Uh,
Sure.
and it's really good that you have spared the time and giving this lecture.
Yeah.
So you can present it. If any problem is there, then we also have the PPT.
Okay. Sure. Sure. I think Ankita's name is showing as a main presenter. So if you can make me the host, I can share it.
Directly you can share, Nasir sir.
You can share.
Yes.
Good morning, sir. You can share directly your own.
Okay.
Yes, sir.
So we'll start after 4 minutes.
Okay. Sure. Sure.
Then.
At 12 we'll start, na? 12.
Yes, yes.
In
12.
the meantime, you
Yes.
can share. Share.
Just please check if you are hearing me and the slides are moving. Just let
Yes,
me
sir.
know.
Yes, sir. You're audible
Yes,
and
yes.
your slides are also visible.
Yeah. Okay. 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 Sure. So we will start after 3 minutes, huh?
Yes, sir. Yes, sir. We'll start after 3 minutes.
Yeah.
I just went through your profile, so you're working on herbal drugs.
Yeah. Sure.
So how is the environment in the Saudi, Dr. Nasir?
Yeah, it's good. It's good. Now we are also start feeling cold.
Acha, okay.
From last 2 days, there is a slight dip in the temperature. Other it was normal. This I think 15 days only there is some cold.
Okay.
Otherwise, the summer is longer than India.
Mhm.
Here it is around
Yeah.
10 months. There is summer.
Only this December and January it will be cold.
Yes, half December to 15th January, you can say. 15th December to 15th January, 1 month. Yeah. I wanted to listen Dr. Ranjit Singh, but I missed that opportunity.
Acha, okay.
Day before yesterday. Yeah,
Yes,
because he was my senior in Bareilly when I was in Rohilkhand University, Bareilly.
Acha, okay.
He was my senior and very dynamic person. Really means I was blessed to have him in my career. At that time, I was having some meeting actually on the day before yesterday, so
Acha, okay.
I missed the opportunity. How was his lecture?
No, his lecture was fine. Very good, speech. He has given very good speech.
We were together around 10 years in Bareilly, Rohilkhand University.
Acha, okay.
Then he moved to Hemwadi Nandan Bahuguna University, Uttarakhand, and I moved to here, Saudi Arabia. And then he switched to Shobit University, Meerut. Nowadays, I think he is in Gango,
And
Saran.
yes. As a vice chancellor,
Yeah, as a vice chancellor. So, should we start or?
Yes, sir, yes. Yes.
yes, you can start.
Yeah, okay.
A very good morning to everyone present here. I, Dr. Ankita Tiwari, on behalf of School of Pharmacy, Sharda University, would like to welcome you back to this third day of 5 days International FDP on the theme Cutting Edge Developments and Future Directions in Pharmaceutical and Health Sciences. It is really our pride to have amongst us our esteemed speaker for today, Professor Nasir Ali Siddiqui, sir. And I welcome him. I extend my heartfelt welcome on behalf of the entire organizing committee. We welcome you, sir. I would also like to welcome all the participants which are there with us on the YouTube live. Thank you for joining us today. And it is really wonderful to see you all here once again. As Albert Einstein said, once you stop learning, you start dying. So let's keep the spirit of learning alive as we continue today. So I would like to briefly introduce Professor Nasir. Professor Nasir Ali Siddiqui is an accomplished academician and researcher in the field of pharmacognosy and phytochemistry with over 23 years of teaching and learning experience. Currently, he serves as a professor in Department of Pharmacognosy at King Saud University, Riyadh, Saudi Arabia, where he has been contributing for more than 13 years. His work focuses on the standardization and quality evaluation of herbal drugs, natural product isolation, and bioassay-guided fractionization, alongside expertise in advanced analytical techniques like HPTLC, HPLC, and NMR spectroscopy. His extensive research portfolio includes numerous funded projects such as investigating the phytochemistry and biological properties of regional plants and developing advanced formulations. Dr. Siddiqui has been involved in various academic and professional roles, including serving on assessment committees, supervising postgraduate theses, and contributing as a member of examination boards in prestigious institutions. He has also served as a consultant and co-investigator on multiple research projects, notably in cancer progression studies and plant-based nano formulations. He has authored an impressive array of publications in very high-impact journals and contributed to significant book chapters, reflecting his deep insights into pharmacological and phytochemical studies. Additionally, he is recognized for his mentorship, having guided master's students in projects on neuroprotection and secondary metabolite characterization. Dr. Siddiqui's contributions to academia and research have been acknowledged through various awards and certificates of appreciation from King Saud University and very other esteemed organizations. His ongoing research reflects a commitment to advancing the understanding of natural products for therapeutic applications. With this, I would like to request Sir for his talk.
Yeah, thanks, Ankita, for my introduction. And I really congratulate all of you for organizing this faculty development program. And as you said, the learning process never ends. So every day we need to learn something new and spread it in the society. As far as my talk is concerned, today I am going to talk on the herbal drugs, how we can maintain the quality of the herbal drugs, and what are the glitches in the quality of herbal drugs, what are the parameters. I think there is some disturbance. What are the parameters we need to consider? And as we know, these herbal drugs are not that easy as we are seeing in case of modern drugs or the synthetic drugs, because there are so many variable factors, the environmental factors, genetic factors, and overall manufacturing aspect, the personnel, the machinery, the methods, what we are applying, then the trade names and common names and vernacular names of the plants. So there are many factors which affect the quality of the herbs. And moreover, when we get in the final product in the market, if you randomly select any 10 products of the same plant, you will hardly find one product is mentioning the real standardization in its label. Real standardization means what? For example, each capsule contains 100 milligram of the root powder of this plant. This is usual conception in the labels. Each tablet contains 500 milligram leaf powder of this. But actually it is not the standardization. And standardization doesn't mean that you are filling 100 milligram root powder in a capsule and giving to the patient, and patient has nothing to do with this powder. So what are the actual content in this 100 milligram? That is more important. This is called standardization. Instead of writing each capsule contains 100 milligram of root powder of this plant, if you are writing each capsule contains 50 microgram of senosides, for example, or 50 milligram of digitoxin. So this is the real standardization. It means you are providing the therapeutic dose to the patient. But in most of the cases, as I said to you, if you randomly select any 10 products, you will find hardly one product writing this kind of information. Each capsule contains 50 microgram of senosides or 50 milligram of the digitoxin. Otherwise, in 9 products, you will find each capsule contains 100 milligram of root powder of Glycyrrhiza glabra or senna leaves or this and that. So these are the means you can say the marketing gimmicks which making the patients or the customers fool, because they are not claiming it will be useful or not, and they are claiming only the quantity of the powder. So it's up to the patient, up to the capsule, up to the product, whether it will benefit or not. So in my talk, inshallah, I will try to focus on what is the quality control parameters and how we can get it a standardized product in our market. So this is my brief introduction. As Ankita told me, I am working in King Saud University, Riyadh, and I joined here in 2009. So around 16 years I have been here. So my topic is quality control of herbal drugs, a futuristic approach, what we can do in future to make the products better. This is the statistics of my university in which I am working, and in Shanghai ranking, my college of pharmacy in which I am working as a professor, it is globally ranked 20th 20th number in global Shanghai ranking.
Sir,
This year it
sorry
was.
to interrupt you. If you could please bring that slideshow.
Okay, sure.
Thank you so much.
So this is my university statistics. As I told you, this year we got the 20th rank all over the world in Shanghai ranking. These are some questions which need to be answered as far as the quality control of herbs are concerned. Like, what are the major issues of standardization of herbal formulation? What are the evaluation parameters? What are the standards applicable to crude drugs and finished products? This is another challenge of the herbal drugs. It is a two-stage analysis, one at the crude drug stage and another at the finished product stage, because sometimes we totally lost the marker compounds in the finished products, what were present in the crude drug form due to some manufacturing processes. Number 4, how we can define standardization of herbal products. What are the marker and biomarker compounds and their importance in the herbal drugs? One is marker compound, another is biomarker compound. How we can encounter the adulteration and assure the safety of herbal formulation. This is one of the major issues currently we are facing. Many products, herbal products, have synthetic contaminants, and everyone dealing with the herbal drugs claiming that it is a miracle drug, it can be useful for this one, it can treat this disease, and basically what they are doing, they are making some adulteration with the synthetic drugs or the modern drugs. Next is, what is the DNA barcoding and how it is related with the QC of herbals. This is one technique which we use in the proper identification of the herbs. Some unique challenges, like it's a two-stage analysis, one at a crude drug stage, another is a finished product. Then changing chemical configuration of the species. As we know, all the plants are a factory of chemicals. So when we process these factory in a manufacturing unit, all these chemicals interact with each other. So sometimes they disappear, sometimes new chemicals are formed. So the chemical configuration is usually it is changing throughout the process. Next is the active constituents variable in the quantity. As we know, they depend on the environment also. So their environment affecting the quantity as well as quality of the chemical constituents. Then sudden appearance and disappearance of negative and positive markers. Negative markers are harmful chemicals which are produced during the manufacturing process or as adulteration. Positive markers are the useful constituents. Then environmental effects, then procedural or technical effects, then personal or human resource aspect. Most of the herbal products available in the market are non-standardized. 1 out of 10 you may find as a standardized product. What are the points which need our consideration? Number 1 is understanding the various stages that crude drug passed before it reaches the pharmaceutical manufacturing line. Then understanding how these stages might impact the nature and amount of active constituents present. Then how to make sure the consistency of active constituents and reproducibility of herbal formulation. It is not like that one batch you are producing very good quality of the herbal formulations, another batch you lost the consistency. Considering the safety and efficacy of herbal formulations as one of the prime objectives and a study of underlying causes and techniques used for assuring the safety and efficacy.
Excuse me, sir. Sorry for interruption again. Sir, your slide is not visible. Could you just click on from the current slide in the slideshow option? Top left side. Top left, sir. From current slide. Yeah. Left side.
Yeah.
From current slide. Yes, sir. From this one.
Okay.
Sir, have you clicked on that?
So, are you seeing the slide?
Yeah, a slide is visible, but I think it's not in slideshow mode. You can even press Control plus F5. That will give you the slideshow view.
Is okay?
Yeah, sir, it's visible.
Okay. So why the quality control is important? These are some factors like cultivation, harvesting, storage, genetics, environment, contamination, preparation, collection. These are the 8 factors, and maybe some other also, which affect the quality of the drug. Quality of the drug here means the chemical constituents which are having the therapeutic potential. So these are the factors which affect the quality of the drug. Then a lot of scope of variation. That is why we need the quality control of herbal drugs. As we know, these herbal drugs are best suited for these diseases like primary health care as a what we call the precautionary measure or prophylactic, like immunity enhancer or something. Then infectious disease like AIDS, opportunistic infection, and multi-drug resistant infections. Then degenerative and gerontological conditions like osteoporosis, chronic arthritis. Then metabolic disorders like diabetes, dyslipidemia, and other conditions like microcirculatory disorder, liver disease, etc. So these are the major areas of the diseases where these herbals are useful. This is the pharmacopoeial definition of the quality control. Like, it focuses on 3 points. One is identity, then purity, then content. As far as the identity is concerned, it is the herb that one should be. It means, for example, I think the example of clove is the best example. The floral bud is the drug, but the stalk, if it is more than 5%, it is considered as adulterant. Again, I am explaining the identity of the plant means the exact part what we need, either it should be or not. If it not, then it is considered as a adulterant. And the best example is clove. Clove, we are generally using the floral buds, dried floral buds. And the stalk included, attached with this floral bud, is considered as a adulterant if it is more than 5% of the clove weight. So this is the specificity of the identity. Then purity. Are there contaminants, like example, in the form of other herbs which should not be there? So what is the herb we are using is pure, or it is mixed with some other what we call the inferior material or deteriorated material. Then content or assay is the content of active constituents within the defined limits. What are the major active ingredients and whether they are within the defined limits or not. If they are below the defined limits, it means it becomes useless. Sorry. These are some classical parameters which we studied in our beginning. If you talk about the 50 years back or 20 years back, these were the basic parameters what we were responsible for the quality control. But nowadays the scene is changed totally, and we are focusing on the chemical moieties. If you talk about the 50 years back, we are considering everything morphological, powder form, this and that. But nowadays the concept is different. If we are using the senocides, plant is whatever it would be, but the senocides needed for the laxative action. So the major active ingredients are the focus area these days, and this we can define by various analytical methods or chemical methods. We can identify the chemical constituents and even quantify, and even after quantification we can grade the drugs. This is A grade, this is B grade, this is C grade. So these are some parameters: organoleptic characters, botanical, physical, chemical, and biological characters. This is the general protocol for standardization of plant drugs. It is starting from here. The raw material is analyzed, then source identification, then proper storage condition, then manufacturing in process analysis, then processed material, then packaging and storage, and finished product. So the analysis, you can see the raw material is analyzed for the constituents, then finished products is analyzed for the stability of those chemicals which are present here. If they are not stable here, it becomes useless to formulate these herbs in any formulation. So we need to analyze at every step to make sure the presence of those chemicals, those ingredients which are responsible for the therapeutic activity. This is the one method how we can reproduce the good herbal formulations. This is one major issue is the establishment of the reproducibility of total chemical constituents. What are the profile of herb when you are collecting in raw form, dried, and making the chemo profiling? So what is that pattern of chemicals in that profile? It should match in the final product. Otherwise, if it is not matching, it means you lost the ingredients. How to establish chemo profiles? This is one most challenging task. This is one preliminary stage testing for the chemical groups. Then quantification of chemical group of interest. For example, if it is alkaloid or glycoside, first you need to identify whether it is there or not. Second, it is quantification of these alkaloids or glycosides. Then according to quantification, you can fix the dose of the formulation. Then fingerprinting of these chemicals with the help of TLC, GLC or HPTLC. Then fingerprinting with the marker compounds. Marker compounds, I think it is characteristic of the herb. It is not related always with the therapeutic effect, but it is characteristic or the signature of the herb. Whenever you are making the fingerprint, these markers are used as a signature of that particular herb. Biomarkers are those compounds which are signature as well as the responsible for the therapeutic activity. Then quantification of these markers. Difficulties in development of chromatographic fingerprint for herbal medicines. These are the some difficulties which we usually face. A large number of chemical compounds.
Dr. Nasir,
Yes.
your slides, just move the slides on. Make it on the slideshow.
Okay. It is there.
Just move the slides also. No?
Sir, if you could please click on that from current slide, or you can even press Shift plus F5. That will.
Shift and F5.
So it is in the slideshow form or not?
No, sir, it's not in the slideshow currently.
Maybe this is some.
Or at the bottom also there's an icon. You can press that, or you can press the keys Shift F5.
Okay, you can move from the left side. Move the slides. Continue, Dr. Nasir.
Okay. So a large number of chemical components are involved in low concentration. The chromatographic instruments and experimental conditions are difficult to reproduce during real analysis. Then the baseline retention time. Then occurrence of abnormal chromatograms. These are the few challenges where we can fight for making a perfect chemo profiling. These are the marker based. These are the marker-based fingerprinting. The chemical markers chemically define constituents or group of constituents of a herbal medicinal product which are of interest for quality control purposes, regardless whether they possess any therapeutic activity. So these are some, as I told you, these chemical markers are the signature of the herbs on what basis we can decide the quality of the herb. If it is present, it means it is a good quality. If it is not present, it means it is a not good quality. Then importance of these chemicals, the chemical markers should be used to various stages of the development and manufacturing of an herbal medicine, such as authentication, differentiation of species, collecting and harvesting, etc. The quantity of a chemical marker can be an indicator of the quality of a herbal medicine. This is simply like when we go to the market and see various type of cardamoms or cloves, and these are the grade, means obviously they are segregated on the basis of their quality. So some cardamom you find A grade, some cardamom you find B grade or C grade. So you decide this you have to take or this you have to take. Same way, the herbs also. When you go to the market, the same herb is you can find in 2-3 grades, and these gradings are based on this quality. The quantity of chemical marker can be an indicator of the quality of an herbal medicine. The systematic investigation using chemical markers may lead to discoveries and development of the new drugs. These are the type of markers. Type of markers, like negative markers means those which are harmful. For example, Aristolochia. Mixing of Aristolochia is very common in Chinese herbal medicines, and this aristolochic acid is very dangerous for our health. Then some active markers are also present, like withanolides in ashwagandha. Then active principles and active markers like senoside and gallic acid. Then analytical markers like resveratrol, rutin, or parthenolide. So these are the examples of markers which you can say that decide the quality of the herb, whether it is good or bad or injurious or harmful or useful or what. This is one example of Chinese herbal formulation in which this is the Aristolochia fungi giving the aristolochic acid. Here you can see the aristolochic acid which is absent in Stephania, okay, and this one. But if it is this Stephania is usually mixed with this one, so here it is aristolochic is absent, but in another slide you will find this is Stephania tetranda, and these are the aristolochic acid you can find in this also. It means in this formulation, this Stephania is mixed with the Aristolochia. So these profiles gives you the exact chemical composition of the formulation and what is mixed, what is pure, and how much it will be useful for you. Application of chemical markers, the identification of adulterants. This is the first application of the chemical markers. Whether your marker compound or biomarker compound is present alone or it is mixed with some other adulterants. So this is the first application of the marker. An adulterant of gamboge Garcinia was differentiated from the 8-K xanthone as chemical authentic sample by an UPLC UV method. Then next use is differentiation of herbal medicines with multiple sources. Multiple sources means, for example, a supplier or a manufacturer is not able to, you can say, fulfill its requirement from one supplier. So usually he take the raw material from different suppliers. And obviously these different suppliers collect their herbs from different regions. So obviously they will be different for their chemical constituents. So these marker compounds, when you develop the chemical profiles, they will differentiate which supplies are good for you and which supplies are bad for you. The determination of the best harvesting time means you can, after analyzing the chemo profiles, you can say this herb is good and at what time it was harvested or what time it was collected, in which season it was collected. So this is another findings related with the chemo profiles. The assessment of processing method, the quality evaluation of herbal parts, identification, and quantitative determination of proprietary products. These are some applications of the chemical markers. Then stability test of proprietary product. Stability test is used to evaluate product quality over time and determine recommended shelf life. It is not like that you can produce a herbal formulation and you can use after 10 years or 5 years. So every constituent has its shelf life. So these are the stability studies which give you an idea. For example, this is a laxative syrup of Senna glycosides. So how long you can use in 1 year, within 2 years or something. So these are the stability studies which gives you the shelf life of the product. Diagnosis of herbal intoxication. What is mixed, what is contamination was given to your herbal product. It is also analyzed with these markers. The toxic components may be used as chemical markers in a screening method. like example, rapid diagnosis of acute head and neck poisoning in urine samples by HPLC-MS. Some lead compounds for new drug delay. Multi-herbal formulations are obtained by subjecting herbal ingredients to various manufacturing processes such as extraction, distillation. These are the common process what we use during the formulation preparation. These are some bottlenecks of the multi-herbal formulations when we are making these formulation. Like every ingredient need to be standardized because it is a multi-ingredient product. The increase of constituents load, disappearance of ingredient characters in formulation, then stability of ingredients versus formulation stability. Individually, how much they are stable, and in formulation, how much they are stable. Inconsistency of finished formulation, large variation in different samples of same formulation, then variation in ingredients and variation in quantity of ingredients, no uniform manufacturing protocols, overlapping of chemical chromatographic profile, stability of formulation, and difficulty in developing multi-herbal formulation. So these are some bottlenecks while preparing the multi-herbal formulation.
So my apologies for interruption again, sir. Could you please change? Yeah, your slides were not visible that time. You can click on the left-hand side, sir.
Yeah, I will do.
Thank you.
Now it is visible?
Yes, sir, it's visible now.
Okay. Here you can see the chemical markers in single herb and in formulation. Sometimes it disappears, sometimes it appears. So this is the fate of the chemical markers. And we need to take precautions whether it is disappearing or it is not. If it is disappearing means your chemical marker is no more in the final formulation. Here you can see this is the formulation and this is the single herb. It is giving a spot here. This formulation, this chemical marker is appearing, but this formulation, it is not appearing. It means we need to change some steps in our processing of final formulation so that it will appear. Otherwise, this becomes useless if this is the main marker compound. Same here, this is the single marker, single herb, and it is missing these markers in these formulations and these formulations also. This marker is missing in these formulation and these formulations also. So sometimes what happens, the single herb shows marker compounds, and when we process the formulation in final product, it disappears. So we need to check at every step where it is disappearing, then we can modify that step. Marker compounds of ingredients, not necessarily the markers for formulation. As I told you, this is maybe they disappear in the final formulation. Some markers undergo transformation and decompose or vanish or regenerated. It depends upon the processing while making some formulation, like sometimes hydrolysis. Like hydrolysis, what it is called, the glycoside is broken down into a glycon plus sugar part. Esters are broken down into acid and alcohol. Amides are broken down in acid and amine. So if you are formulating a glycoside, you need to make some changes so that the glycosidal state must be intact during your processing. Otherwise, it is of no use. So it means when you analyze or you make the chemo profiles of the crude drug, you will see the glycoside. But when you make the chemo profiles of the formulation, there will be no more glycosides because it is broken into glycoside, a glycon and sugar. So these are some steps which convert your original chemical markers into some other ingredients like hydrolysis, dehydration, esterification, glycosidation, isomerization, and rearrangements. So we need to check at which step it is disappearing, and we need to modify those steps. New markers or marker compounds specific to multi-herbal formulations need to be isolated. If new compounds are formed, then we can isolate and study. The formation of new markers depends on the various manufacturing steps. The multi-herbal formulation multi-herbal formulations marker compound. This is new classification. Like chemically modified markers are those markers generated by dealkylation, deacetylation, dehydration, and isomerization. It means any of these steps, if it is happen during manufacturing, these marker compounds are called chemically modified markers. Then hybrid markers produced by condensation, esterification, glycosylation. Pseudo markers formed by degradation, decomposition, polymerization. Impurity markers, these are negative markers formed by pyrolysis. Pyrolysis means whenever heat is applied to them for the formulation, like in bhasma or chara or lavana. In Ayurvedic preparations, we use heating. So these impurity markers may appear. Then markers quantification. The markers may serve to calculate the dose quantity of herbal drugs in the finished product if that marker has been quantitatively determined in the herbal drug when the starting materials are tested. These marker compounds are quantified and the grade of the drug is decided on this quantification. Chemical fingerprint analysis method. How we can make the chemo profiles. These are some techniques used for that, like thin layer chromatography, gas chromatography, HPLC, HPTLC, capillary electrophoresis, and spectroscopic techniques. First, we discuss the HPTLC. HPTLC is a sophisticated and automated form of TLC based on validated methods and globally accepted, important, essential, and viable tool employed in process development. This HPTLC is a technique you can do every study. Just you need to, the most difficult step of this technique is to make the mobile phase. Once you are able to design the mobile phase and your compounds or the ingredients are running on TLC, then you can do any analysis. You can quantify, you can derivatize, you can do anything. So this HPTLC is very useful technique. Simply it is an automated TLC, you can say. In TLC you are doing everything manual, and HPTLC it is automated. Everything is done by machine and you can do any analysis with this one. The only challenging task is to develop a suitable mobile phase. Here I have taken the example of Shank Pushpi, and as you know, the Shank Pushpi is a common name, and the common name Shank Pushpi is used for many herbs. Here I have taken the 4 herbs like Convolvulus pluricolis, Evolvulus alsinoides, Clitoria ternatia, Conscora decussata. These are the 4 herbs which commonly called as Shank Pushpi. So Shank Pushpi, as you know, it is used as a brain tonic. But how you can say that Convolvulus is better than Evolvulus or Clitoria is better than Conscora, and whether they all have the same ingredient or not? So we did one study with HPLC also and with HPTLC also to make the comparison of the ingredient. What are the chemicals common in these 4 plants? Here is the HPTLC study. So you see, this is the Convolvulus pluricolis. This is Evolvulus alsinoides. This is Conscora decussata. This is one brain tonic. This is Shank Pushpi syrup. This is one tablet. Okay. So here you see this peaks. 2 peaks are common in all. This one and this one and this one, this one. So it means this Convolvulus pluricolis, which is called Choisy or the Shank Pushpi, the most of the authors or the scientists, this plant are recognized as a Shank Pushpi. Otherwise, others are less popular. So these 2 peaks are common.
So could you?
Yeah. These 2 peaks are common in all these. It means these 2 chemicals are common in all the 4 plants, which are making the basis of the their common names, you can say. Every plant is called as Shank Pushpi because of they have the same effect Why? Because they have the same chemicals. Same way, the HPLC also study. Here you can see this is the HPLC chromatogram of Evolvulus alsinoides, Conscora decussata, Clitoria ternatia, brain tonic, Shankpushpi syrup, and Convolvulus pleuricolis. So you see these peaks. This is exactly almost matching with this one. Evolvulus alsinoides is exactly matching with the Shankpushpi syrup. This Convolvulus pleuricolis is exactly matching with the Clitoria ternatia. Conscora decussata is exactly matching with the brain tonic. So, but this peak is common in almost all the chromatograms. This peak. It means that is on the basis of this chemical only. This chemical only, all these four plants are commonly called as Shankpushpi. And because of this chemical, they have the same therapeutic potential or the same use as a brain tonic. Now we are coming to this new technique of DNA barcoding. It is called as GPS for taxonomist. What we say, the like these days we are using GPS for reaching to any new place, and we can easily reach to our destination through this GPS. The same way the taxonomists are using these DNA barcoding to identify exactly what is the genus. And the NCBI data, if you give any DNA barcode to NCBI data, they 100% they can give you the correct genus. And if you can provide more information, they can give you the species also, which species it belongs. So this is the best technique till now what we can use for the identification, because identification is a very critical in herbal drugs. Sometimes you might have heard many disasters in during for this identification. In America, I think 1907 or something, there was an incident in which 300 people were died. They collected, misidentified a plant, and there was a one tea in the market used for weight loss, and that plant was misidentified, and it was having a cholinergic alkaloid, and it was processed and marketed, and it was available in the market, so the customers used that one, and 300 people were died. So identification is one of the most important starting point of this herbal research. So this DNA, what is DNA barcoding? If you take any small piece of plant and extract the DNA near about 400 to 500 base pairs, and this base pairs is DNA base pair is called as barcode, you can see, because this is a unique for this genus and the species, and these barcodes are indicating towards some genetic information of the plant. These genetic information is used for the identification, particularly those DNA fragments which are very stable and hardly it is changing something, because in the plant you can take the DNA from the nucleus, from the mitochondria, from the chloroplast. So as for now, that the studies have revealed that the chloroplast DNA is most stable DNA, and any fraction, small fraction of this chloroplast, if you study this DNA and generate a barcode of 400 or 500 or 600 base pairs, this will be the exact giving the identity of the plant, genus as well as the species. So DNA barcoding is regarded as an efficient tool for herbal medicine identification. Single locus or multiple loci have been widely used and have provided sufficient resolution for the identification of most herbs. It is the current main molecular identification method, but still has some limitations. The DNA barcoding is a standardized approach to identify plants and animals by minimal sequence of DNA called DNA barcodes. The DNA short sequence are called as DNA barcodes ranging from 400 to 800 base pairs. DNA barcode, it is a short gene sequence taken from a standardized portion of the genome that is used to identify species, match with the references. As I told you, NCBI has a good data of DNA barcodes, and if you provide them the information, they can give you the exact genus and the exact species. And sometimes if it is not matching with their data, it means it is a new species. So this is another application of the DNA barcode, that it helps in identifying the new species. The use of DNA sequences for species identification has a long history, but it is received in significant attention only after it was formally proposed as a DNA barcoding in 2003 by Dr. Paul Hebert. He called as father of DNA barcoding. He is a Canadian scientist, and he started this method. What is the successful DNA barcode? It is having, these are the characters of the successful barcode. One is short enough to be quickly sequenced. As I told you, because DNA is a long chain, maybe thousands of base pairs involved. But here we are in a specific portion, we need 500 to 700 base pairs. Easily identified in all species of organisms. Variable enough to provide a unique sequence for each species. The size of sequence is 600 to 700 base pairs, and it is a universal character. Which gene fragments are used? Usually in prokaryotes, ribosomal RNA genes used for species identification. In plants, RBCL or ribulose biphosphate carboxylase gene sequence and MATK gene sequence of chloroplast are used. In fungi, the internal transcribed spacer or ITS region is used for the DNA barcoding. This is the processing of the DNA barcoding. This is plant material, then extraction of the DNA, and then studying of the providing the this DNA base pairs. Every time. The barcode benefits. What are the benefits of this barcode? It is a taxonomic identification tool, alternative or additional to morphology. Obviously, the morphological characters or the anatomical characters are taken into consideration while identification, but this barcode is additional, you can say the information is giving. The DNA sequencing is a rapid and relatively low cost technique. It can process a great number of specimens at a time. This is useful, for example, in biodiversity surveys. Once reference database is established, it can be applied by non-specialists, because once database is established, then no scientist is needed for that one. Any person can easily use it. The applications of barcode, the first is the identification of established herbal raw material. Then if it is not matching, as I told you, if it is not matching with the database, it means it is a new species. So discovery of new species is another application of the barcode. Then protection of endangered species. The eliminating species can be protected. Then disease diagnosis. Example, in herbal raw material or veterinary parasitology, it is used for the disease diagnosis. Then pest diagnostics in agriculture or in food, farming science and herbal drug cultivation. What type of pests are harming the crop, it can also be identified with this barcode technique. So these are the other applications like conservation and biodiversity studies, then conservation and biodiversity studies, identification and authentication, then species delimitation, quality control, forensic applications. Forensic application means the if some traits are of illicit drugs are made, then only these barcodings are used for the identification of the illicit drugs. Then global trade felicitation, rapid assessment of raw material, then traditional knowledge verification. In herbal drugs application, where we are using these drugs, the medicinal plants for which nucleotide signature have been used for the identification include Crocus sativus or saffron, then ephedra, salvia, angelica sinensis, ligularia. These are the plants on which it is applied till now for manufacturing, for identification, and for quality control. To detect the toxic additive of Aristolochia, distinction between two species. Sometimes we have 2-3 species and we don't know which species is better for as a drug. So we can differentiate these species on the basis of their chemical markers. Then we can correlate with the DNA barcode, like curcuma longa and curcuma aromatica, Panax quinquefolius and Panax notoginseng, Theobroma cocoa and Theobroma grandiflorum, Senna obtusifolia and Senna occidentalis. So these are the species which can be differentiated on the basis of the DNA barcodes. In China, the 40 herbal dietary supplements labeled with the ginkgo biloba tested for quality on the basis of the DNA barcode, and it is found only 14 were the genuine out of this 40. Other than this 14, all are having some adulteration or contamination with the ginseng. These are the stages of studying, how we can study, how we can proceed. Number 1 is the collection of the sample. Number 2 is the identification and herbal deposition by the taxonomist. And then this is the DNA extraction, and after extraction, development of the profiling. It is called RAPD profiling or SCAR profiling. RAPD, there are certain markers. These are the markers RAPD, SCAR profiling, which can be used for the identification purposes. Step number 5, DNA sequencing and then DNA editing. Like here you can see these are the DNA base pairs, adenine, guanine, thymine, cytosine. So these are the sequence editing, and these what we call the DNA barcodes are made on the basis of these sequences. These are the DNA barcodes. On that basis we can identify the particular genus and then particular species. Here you can see the diagrammatic representation of SCAR marker development for genuine and adulterant samples using RAPD profile. This is the profile. These are the base pair 10,000, 8,000, 250, and this is the marker compound. This is genuine sample. Lane 1 is genuine sample. These are the adulterated sample 2, 3 and 4. So you will find this marker is missing in sample 2, sample 3, sample 4. It means these are the adulterated, not having this marker compound. RAPD profiling for genuine and adulterant sample. Lane M marker. These are the markers. And then lane 1 is genuine sample. This is lane 1. Here is the genuine sample which is having this marker. Lane 2, 3 and 4 are adulterant samples. Lane 2, 3 and 4 are adulterant sample, because in this lane, 3 lanes, this marker is totally missing. This is the information what we can get from the DNA barcoding. This is another sample, SCAR marker for genuine sample. Lane 1, This is the genuine sample. And band absent in lane 2, 3 and 4. This is lane 2, 3 and 4. This marker is totally missing in these adulterants. So again, we can say that these are the adulterants and this is the genuine sample. These are some international organizations which are working on DNA barcoding. Consortium for the Barcode of Life and European Consortium for the Barcode of Life, and many others also. It is international initiative taken in 2004 to develop DNA barcoding as a global standard for the identification of biological species. 200 member organizations from 50 countries are working on this DNA barcoding. Then European Consortium for the Barcode of Life, established as part of the research infrastructure efforts of EDIT and European Distributed Institute of Taxonomy. This is International Barcode of Life, then Canadian Center for DNA Barcoding, largest biodiversity genomics initiative, 15 nations working together to construct DNA reference library. Around 500,000 species are decoded for this one. Then Canadian Center for DNA Barcoding is largest barcode factory generating hundreds of thousands of data records per year, training barcode researchers from around the world. So these are some organizations working on DNA barcodes. This is some list of plants with their adulterants identified by DNA barcoding. Other than this, there are many more plants, but I selected only few just as an example. Like Artemisia, adulteration is Artemisia capillaris, and this is the technique marker use. Scar markers were used for the identification. Panax quinquefolius, these are the adulterants. This technique is used for the DNA barcode. Phyllanthus umbilica, amla, other Phyllanthus species as adulterant. Pureria and Ipomia as adulterant. Curcuma with other varieties. The Astragalus root, and this is the adulterant. Senna species and different other Senna species. Low content of senosides. RAPD is used for the differentiation. Cuscuta with this adulterant, Ruta graveolens with this, Euphorbia, Glycyrrhiza with Abrus precatorius, and Dendrobium with Dendrobium species. So these are the main herbal drugs, and these are the main adulterants which have the almost similar morphological characters, similar powder characteristics, but they have lesser active ingredients. So these are the used as adulterant, and we can identify with these techniques of DNA barcoding to differentiate these two. So what is the conclusion of our talk is the quality control of herbal drugs and formulations required all the good practices. You can say the quality of good herbal drugs can be obtained by good agricultural practices, good manufacturing practices, good clinical practices, good hospital harvesting practices. All these good make your quality or the herbal formulation good, you can say. The use of analytical methods to establish the presence of biomarkers in crude drugs as well as formulations and assure the therapeutic efficacy. This is most important point. The quantitative determination of biomarker in the final product is needed for a particular for a standardized preparation. Use of analytical method to detect the presence of adulterants in crude drugs as well as formulations and assure the safety of the herbal formulation. This is another point. We can make our herbal formulation safe by checking the adulterants. HPLC, HPTLC, GCMS and some other techniques are preferred techniques used for the quality control. Then use of DNA barcoding to establish the identity of good quality herbs. This is one additional tool we can use for identification and differentiation of the good and bad species. Use of DNA-based molecular markers to establish the purity of herbs and surety of therapeutic efficacy. So by this way, we can make our herbal formulations as perfect as possible or as efficacious and safe as possible by applying all these parameters. So thanks to the organizers for giving me an opportunity to discuss this talk about the herbal drugs and formulations. And if you have any questions or query, please, you are most welcome.
Thank you, Dr. Nasir, for giving such a wonderful presentation. And I think you have touched all the aspects of the quality control and the standardization. That is the quality control markers, then the HPTLC techniques, and the DNA barcoding especially, which is a very new thing for us. And I think it will be very beneficial for all of us and all the delegates. These kind of techniques, it will be very useful to them. Okay, thank you once again.
Yeah, thank you.
Yes.
Thank you so much, sir, for such an enlightening session. And we are incredibly grateful for the time and effort you have dedicated for sharing such an important topic with us. Among the chat box, I can find a question from Mr. Vijay Nigam. He is asking for new crude drugs applied for research in either MPharm or PhD, where we should go for the DNA barcoding. So if you could please answer his question.
Yeah, sure. The new species, it means that nothing has been done on before on that particular herb. That is called new, as far as my knowledge is concerned. So the first thing is you need to go to literature. If something has been done not in the as a therapeutic effect or some other aspect like agricultural aspect or other things, you can find something on that particular herb what you are selecting, and you are saying it's a new. So if there is no literature, then you can start the work on that one. So literature is one tool what you can consider for your herb if you are going to start for the first time and nothing has been done till now. This is one aspect. Another aspect is if you have the problem of identification, then we can go for the DNA barcoding. If some taxonomist is not able to identify this herb, then you can go for that one. And I don't know in India where you can go for this DNA barcoding. Maybe in NBRI Lucknow they are doing probably, or CDRI Lucknow. But if nothing has been done, you can this give me the samples. I can give you the DNA barcode for that one, and I can give you the identification tools. So I hope I answered your query.
Yes, sir. Very nicely you have answered his query. Thank you so much, sir. I would like to thank all the participants who have joined us online YouTube live for this FDP. And with this, we come to the end of day 3 of our session. And thank you for your kind patience and listening. And for tomorrow, we have our speaker from Deminar University, Professor Gabir Betta. He is from Egypt. So I request you all to be there with us tomorrow. The link will be shared on the WhatsApp group. Thank you so much for your time. Have a very nice day.
Okay, thanks a lot.
Thank you, sir.
Thank you, Dr. Nasir. Thank you so much, sir.